Go. Population genetics. This discipline comes from this time period in the early 1900s that we call the modern synthesis. So before this time, scientists were really working in their own little pockets, their own disciplines. So mathematicians were just working with mathematicians. Evolutionary biologists were really focused on what they were doing. Geneticists, microbiologists, everyone was sort of working separately. During the modern synthesis, scientists start to kind of collaborate, to think about ways that their disciplines overlap. And so out comes this idea of population genetics, where we can quantify changes in allele frequencies from one generation to the next, which is, as you know, the definition for evolution. So we are really thinking about how we can quantitatively describe this process of evolution. We should get really clear on what a population is. We discussed this in the context of evolution by natural selection, but let's talk about it again. A population is a group of individuals. They are the same species, which means they can interbreed, and we think of them as more or less isolated from other populations. So we can think about a population and the dynamics, the things that are happening among those individuals in that population, separate from other populations. So this little diagram is showing us that we've got maybe an original population, and then some move to one island and some move to the other island, and that gives us two populations. Okay. So again, population genetics, we're thinking about the same processes that we talked about with regular genetics, individual genetics, but now we're thinking, instead of the alleles that one individual or two individuals have, we're thinking about the allele frequencies and the genotype frequencies in a population. So we're studying forces that can alter the amount of genetic variation in a population. Remember that your genetics, my genetics, the mice's genetics don't change in their lifetime. What changes is the population, the relative frequencies of different alleles. So that's what we're thinking about here in population genetics. And we've got some really useful mathematical tools to do this. So with population genetics, I'm hyping it up, trying to get you excited. What can we do? We can ask questions, interesting questions like, is this population evolving? If it's evolving, how is it evolving? Are some genotypes favored or disfavored? How quickly is the population evolving? How strong is selection for or against certain genotypes? So we can really, don't write these questions down. We can really answer and ask some interesting questions with population genetics. So let's start off with a population that's not evolving, so you can start to see how we're going to do this. So on the right, got a population at one point in time. On the left, we're thinking about that population at a different point in time. And I'm displaying each of these circle individuals' genotypes in letters inside the circle. So you see we've got some homozygous dominant individuals, some homozygous recessive individuals, some heterozygotes, right? And if we counted up all the big G's and the little g's, you would determine that there is a frequency of big G's that is 37.5. So 37.5% of the alleles in this blue box are big G alleles, which leaves 62.5% as little g's. Okay. In our second box, later in time, we could count up the alleles and find the same frequency. So the allele frequency has not changed. So evolution has not happened, but you can see that we've got more individuals over here. I'm worried that this diagram might be wrong. I might have made a mistake. Point is that allele frequencies can stay the same, but genotype frequencies might differ. We might have more, or in this case, fewer homozygous dominance and more heterozygotes, even having the same allele frequency. Okay, so this is a population that's not evolving. The allele frequencies didn't change. How did we get the individuals in this second blue box? We got them by random matings in this first blue box. All right. So if we want to ask questions about change, we first need to establish a baseline, our expectations. What do we think the population is going to look like if no change is happening? So this is kind of like our negative control when we talked about experimentation. We need to decide, or we need to determine what we think the population will look like if no change is happening, so that we can see if change is happening or has happened. Okay. So one product of that modern synthesis I was talking about is the principle that underlies this area of population genetics. And that principle is the Hardy-Weinberg principle, named after Hardy and Weinberg. They were both mathematicians, scientists, and they independently came up with this idea. So what this is a null hypothesis. This is what we expect if evolution is not occurring in the population. So this defines our expectations for a population that isn't evolving. So we can compare this expectation to what we actually observe to see if our population has evolved or is evolving. All right, so here's a metaphor that I think is going to help. Let's say we have a coin, or maybe a group of coins, and we flip, let's think about one coin, and we flip it however many times we want. How would you decide whether that coin is fair, whether it is giving you your expectation? You're thinking about this, and you're probably thinking, well, if I wanted to determine if the coin was fair, I would flip it a whole bunch of times. And I expect that when I flip it a bunch of times, I'm going to get half of the time heads and half of the time tails. So we expect something like this. And we know that we would need to flip it more than 200 times to approach something that's perfectly 50-50. But these data would probably satisfy us that our coin is fair. All right, so this is like establishing our baseline. We expect 50-50 heads and tails. We flip our coin and we see what we find. If we got data like this, we would say, hey, there's something wrong with our coin. Maybe it's weighted on one side. Maybe it's damaged. This coin has been tampered with. We know that something is not right. Okay, this is the kind of thing we're doing with population genetics. We have to establish what we expect our population to look like and then compare what we actually see to that expectation. Another concept we need to get out of the way, and I don't think this will be a total revelation for you, is this idea of a gene pool. You can think about this like a bag of beads, like we saw in lab. So the gene pool is all of the alleles for all of the loci for all of the individuals in the population. And for our purposes, we are going to assume that within this gene pool, fertilization or union of gametes is random. Any gamete has an equal likelihood of fusing with any other gamete. So this is just like reaching into your bag of equally sized, equally textured beads, grabbing two. The one you grab first has no bearing on what you grab second. Okay, so random fertilization. And we've talked about this before when we talked about meiosis. All right, and in our gene pool, for our purposes, we're just going to ever think about one gene and that gene with two alleles. Okay, so here's back our non-evolving population, and in the lower right-hand corner, there's a Punnett square. So we have been thinking about genetics, sort of like two individuals mating. Now we're thinking about the whole population on one side of our Punnett square and the whole population on the other side of the Punnett square. We can get our first gamete, and then we're going to get our second gamete. And the only thing that controls the likelihood of getting one allele or the other is the prevalence of that allele in the population. So if we have 37.5% big G's, we think 37.5% of the time when we reach into our bag of alleles, we're going to get a big G, and 62.5% of the time we're going to get a little g. Okay, and the same thing when we reach in the second time. And what we know is that since we're doing two things and we're doing them both together, we're using our multiplication rule, so the likelihood that we reach in and get a big G and reach in and get a second big G is 14%. That we get a little g and a little g again, 39%. Okay, so this is just the same idea as our genetics, but instead of having in your individual, in your one of your parents, 50% big G's and 50% little g's because they just have the two alleles in a diploid individual, instead of our known, like our coin, heads and tails, 50-50, we have maybe different quantities because we're thinking about our whole population on one side of the Punnett square and our whole population on the other side of our Punnett square. Right, so similar thoughts, but a little bit different framing. All right, so we can, based on the composition of the population, we can decide what we expect in terms of our different offspring genotype frequencies. And we're going to look at this in more detail as we go on. All right, the last thing I want you to think about before this video stops is the life cycle of a population. We've talked about the life cycle of an individual population. There's really nothing new here, but we're just scaling up. So we've got lots of adults undergoing meiosis to make gametes, our whole gene pool there. Within our gene pool, random fertilization. Zygotes are created. They grow up into juveniles, and juveniles grow up into adults. And if no outside forces, if no evolution is happening in this population, we just expect to spin around and around and around this life cycle with no changes in allele frequencies. Okay, so we're going to get to this idea of establishing a baseline. Okay, I'm just checking the time. So this is the equation. I just want to show it to you now. It's going to come up later. P squared plus 2pq plus q squared equals 1. This summarizes what we expect for a population that's not evolving. This is not a plug and chug situation. Okay, it's always going to equal 1. And I think I'm almost out of time, so the last thing I want you to write down is that we think of P as equal to the frequency of the dominant allele and Q as equal to the frequency of the recessive allele. These are things we can measure, and we'll use them as we go forward. All right, so I want you to take the assessment on CourseConnect. Your grade doesn't matter, but the fact that you do it does matter. It's going to test you on these 3 things. Once you've taken assessment number 1, come back and watch the second video where we're really going to dive into how do we do population genetics? What's the logic behind this idea? Okay, so I'll see you soon.